Thanks, Prof. Niveen. Next speaker, Prof. Ahmed Mustafa, Professor Pathology and Lab Medicine, HLA Director in University Saskatchewan in Canada. Professor Ahmed, he will talk Epitope mismatch as a tool for prediction of graft outcome. تفضل دكتور.
First, I'd like to thank Professor Dr. Muhammad Salah Zaki for his kind invitation. And I would like to thank the organizer as well for the awesome conference. So I just added this slide to get an idea about the audience. So how many transplant nephrologists, can you raise your hand, please, here? Okay. Is there any surgeon attending? I'm guessing the answer is no. Yeah, yes. Anyone from the lab are attending the session? Oh. Excellent. Okay, so how many of the transplant nephrologists and surgeon consult with the lab before the process of transplantation? Does anyone consult with the lab directors or the supervisor before transplant? Like when I say consult, do they not blame them about a result, but do you actually consult? Okay, excellent. So in this question here, who is to blame if the patient's starting to reject the kidney? Is it the lab? Are we going to blame the lab if the patient rejects the kidney? Or the patient himself? Or are we going to blame the nurses or the transplant surgeon or the transplant physician? Anyone have any clue here? Or all of the above? Actually, that is the correct system. None of the above will be blamed, and I will let you know why. So before any process of transplantation, there is a committee is formed. This committee is formed of a transplant nephrologist, a transplant surgeon, a transplant nurse coordinator, and a donor coordinator, and the lab as well is involved. So it's not a one-man show. It's usually a decision made by a committee. This committee usually meets twice a month for listing for live donor, and during urgent when there is a diseased donor is on the list. And this committee is not something we recently know. This committee is actually in the history. So this is a picture of the first transplant in the history, and you can see that there is a surgeon, there is someone from medicine, there is the nurse coordinator, and here is the lab director as well. So how did the field of HLA started? So the field started in 1958 by the Nobel Prize winner John Dosey. When he was in the lab and he was very bored at this time, he asked the nurse to bring him some blood. So he isolated the white blood cells from the blood, and he started to put the white blood cells on a piece of slide. And then he asked the nurse to bring him a serum from transfused people, multiple transfused people. And he took the serum. And at this time, he didn't know that this serum contained antibodies. And he started to play with the antibodies or the serum with the cells. And then he looked under the microscope, and he see that sometimes the cells get agglutinated and sometimes not. But what he actually interesting noticed that the patient's own serum does not agglutinate his own cells. And sometimes it agglutinates and sometimes it doesn't. And he discovered the first HLA gene, which we know now as the HLA-A2. So what does human leukocyte antigen stand for? So human, it comes from human, and leukocyte, because as I mentioned, all the initial experiment which was done on white blood cells, and antigen because it's a specific markers expressed on the cell surface. And when I mentioned the word antigen, then we have to know that there is an immune response in the form of antibodies. So how does the HLA antibodies are named? Usually HLA antibodies are named according to the antigen that it binds to. So for instance, this one is going to bind to A1. So this is an HLA-A1 antibody. This one is going to bind to the DR7. Then this is an HLA-DR7 antibody. This one is going to bind to the CW9. So this is an anti-HLA CW9 antibody. But actually, I lied on you now. So antibody never bind to antigen. Antibody bind to something called epitopes. So if you have a picture here, this is a picture of a traditional HLA class 1. The traditional HLA class 1 express something called epitopes. So it's the same idea of the lock and the key. So if this antibody is going to bind, it's going to bind to the best fit epitopes that can bind to. So why this is important? So for example, if you have 4 different HLA genes, as in this slide here, you can see there is an HLA-A2, HLA-23, HLA-24, and HLA-68. Each one of these symbols represent an epitope. So if there is an antibody going to bind to HLA-A2, the same antibody will recognize the HLA-23 and the HLA-24. And this is what we call the cross-reactive group. The same scenario will happen if this bind to the purple oval, it will bind to the 24, and so on, and so on, and so on. And then there is also what we called allele-specific antibodies. And this is the antibodies that is going to bind to specific alleles. So in the clinical field, why it's important to know this cross-reactive group? So for example, here, if we have a donor who is A3 and B51 and has a recipient who is A3 and A31, B51 and B8, so they are actually mismatched at the A2 and the B7. So once this patient going to lose the kidney, then he have a sensitizing antigen in the form of A2 and B7. So, but basically, because of the cross-reactive group, he will form antibodies against all this, what we call A2 cross-reactive group. And also he's going to form all these antibodies against the B7 CREG. And this will result in a CPRA of 88%. So just from 2 single antigen, the patient become a highly sensitized patient and only have a 12% chance to find another donor. So that's why it's important to do pre-risk transplant risk assessment before transplantation. So here I added another example of the epitopes. So here you can see this is the patient HLA typing formed of, just for simplicity, I added 4 different epitopes. And there are 3 different donors or 3 different live donors that we want to pick one of them. So, and the same picture could be switched or reversed if we're going to use a diseased donor with multiple recipient. So for example, here, the first one has a 4 epitope mismatch. The second one has only 1 matching, and then there are 3 mismatch. And the third one has matching at the 3 epitope and have 1 mismatch. So if you look at this at the epitope level, you can appreciate that donor C is the best fit for this patient. And here is another example of how the HLA matchmaker work. So this is basically a software that we use in the lab. This software is free. It's available for anyone to use. So if for example, here, if we have a donor who is HLA B18, and this HLA B18 has 11 different epitopes. And now we have a patient who is B7, but none of the epitopes of the B18 matching the patient. So basically, the patient will develop 11 different antibodies against the 11 different epitopes of the B18. But if we look at the whole picture, you can imagine that this is not going to be the case here. So the patient is typed as B52 and A33. So if you look at the combination of the 3 alleles, you can see that the patient is actually matching the donor. So if you put the B52 with the A33, the 11 epitopes the patient has. So the patient will have this considered as a self-epitopes. So although they are mismatched at the antigen level, they will never form an antibodies at the epitope level. So this will be the best fit for this patient because there were no antibodies is going to form. And this is how we write the applets. So that you can see there is a bunch of numbers and letters. So the numbers represent the location of the amino acid in the antigen, and the letters represent the amino acids. For example, T stands for threonine, L stands for leucine, and R stands for arginine. And you can see here it's number 80 and 82 and 83, and one is skipped, and this is what we call discontinuous because the antibody bind to the conformational change of the antigen. Down here you can see an example of a linear amino acid 81A, 82, and 83. And also epitope can be just form of 2 amino acids or could be form of just 1 amino acid. And this is another software that the lab can use or anyone use, because I know that high resolution typing is very costly in Egypt. But if you can use this program, you can put your low resolution typing and you can select the ethnicity of the patient, and actually it can predict and hit the submit button. It can actually predict the high resolution typing of this patient. And I have used it, it's very accurate. So this is the software that we use in the lab. So in the first column here, I added the patient 4 times, and I added 4 different donors. And what does the software do? It actually compared the patient typing with the 4 different donors, and will tell you how many eplet mismatch is present between the donor and the recipient. And you can see here that donor 3 has 7 eplet mismatch. So if we are going to pick one of them, then donor 3 will be the best fit for this patient. And this is how our the report that comes from my lab. You can see at the top we put the patient typing, and in the bottom we put the donor typing, and then at the last line here we put the number of mismatches between the donor and the recipient. So for example, if we look at the A1 and the A26, and the donor is 24 and 68, so this is 2 antigen mismatch, and so on. But actually, when we put the eplet mismatch, we put the eplet mismatch also in the report, you can see that the numbers have changed. What I found actually very interesting is this one here. So although the DR beta 1 are mismatched at the antigen level, but they are matched at the epitope or the eplet level. And this research has done by one of my friend, Chris Webel from University of Manitoba. It's a retrospective study done on 600 patients, and he grouped them into 4 groups. The group 1 was 1 DR mismatch, group 2, 2 DR mismatch, group 3, 1 DQ mismatch, and group 4 is 2 DQ mismatch. And each dot that you see here represent 1 patient. And he started to count the number of eplets in all of these patients. And you can see that the number of eplets varies from 0 up to 40 eplets with each group. But if we took an example of these 2 cases here, although they have 1 DR mismatch, you can see they have almost 33 eplet mismatch. But if you look at the other one, which has 2 DR mismatch, they only have 10 eplet mismatch. So if we are going to put the immunological risk, then for sure, although they are 2 DR mismatch, they will have a better graft outcome. And he started to do what we call a cutoff. And from the cutoff, he recognized that patient who has up to 11 eplet mismatch behave very well as they are the zero mismatch. But if you have more than 11 eplet mismatch, then there is a higher risk of graft loss. Someone may come and ask me, eplet mismatch is not the only predictive value. Yes, it's not. And also one of the important factors is the adherence, if the patient is taking the medication or not. So Chris again used this electric tool bottle cap and give it to 195 patients. So every time the patient opened the cap, it signals, so they can know if this patient is taking their medication or not. And by this time, they put two groups, people who were not adherent and people who were adherent to the therapy. And you can see here that patients who were non-adherent had higher risk of graft loss. But this was not the idea of this research. The idea was to add the eplet mismatch to this. So what he did, he subgrouped this patient into 4 different groups. Adherent with eplet mismatch less than 10, adherent with eplet mismatch more than 10, non-adherent with eplet mismatch less than 10, and non-adherent with eplet mismatch more than 10. And you can see the worst group were the people who were both non-adherent and have eplet mismatch more than 10. The significance of this, if you did it to your patient, the patient has to know that they are very high risk. So even if they missed one medication, there are a high risk that they will lose their graft. Versus the patient who are less than eplet mismatch, if they lost one medication, there is still a chance to keep the graft healthy. And this is one of the famous study. It's called the CT09. And in this study, they were trying to identify a specific group which they call the immune quiescent kidney. And this group, they want to use it to eliminate immunosuppressive from them. So basically, give them the kidney and give them the minimal immune suppressor, or at least remove the tacrolimus because of the side effects. So they picked 52 patients. This patient didn't have DSA. Their PRA were less than 30%. They gave them the high-end treatment at this time, which is the thymo, the tacrolimus, the MMF, and the prednisone. They were monitoring them every month for 6 months, even doing protocol biopsy. And after this, they were able to identify 2 groups. The group 1, 7 patients who took the 3 treatment, and the other group 14, which they removed the tacrolimus from the medication. But unfortunately, this trial was stopped because all these patients who started to take off the tacrolimus, they started to develop some sort of rejection, either T-cell mediated rejection or antibody mediated rejection. But what Chris did is he actually was interested to look at the eplet mismatch. So what you see here, the patient who lost their kidney, they have an eplet mismatch more than 17. The patient who did not lose their kidney had an eplet mismatch less than 17. So even if you could use this eplet mismatch, you can identify patients who are very low immune compromise. The other thing which was very interesting, even this patient who has a higher eplet mismatch, after they stopped the trial, they developed a donor-specific antibody. And this one developed T-cell mediated rejection. So in conclusion, epitope mismatch can replace the traditional antigen mismatch for predicting donor-specific antibody development, rejection, and graft survival. Epitope matching may be a useful tool for organ allocation and post-transplant care decision, especially if we are going to go with the disease donor. And also it could be used as a personalized medicine for adjusting the dose of tacrolimus. So in our center, we use it for adjusting the tacrolimus dose. And here, this is just like a figure represent the evolution when we start with the agglutination and ended by the HLA matchmaker that we are using now. I know that most of the lab are still sticking with the CDC and the flow, but it's time now to evolve. At the end, I would like to thank again the organizer, and I would like to thank Dr. Muhammad Salah for this nice invitation, and I will be happy to answer all your questions. Thank you so much.
Thank you so much for this elegant presentation. I would like to end this session and invite the panelists to the discussion.